This video will outline the steps for one flask over the duration of one time point. Now that the working environment is sterile, we can now proceed with the preparation. Collect the appropriate number of microcentrifuge tubes for the given time point. For this time point demonstration, you will need 6 tubes, 1 empty, 4 containing 900 microliters of LB medium, and 1 containing 990 microliters of LB medium. You need to label the tubes. You should label the empty tube with stock, the tubes containing 900 microliters of LB medium with 10 to the first, 10 to the fourth, 10 to the fifth, and 10 to the sixth, and then the tube containing 990 microliters of LB medium should be labeled 10 to the third. In order to differentiate between the plates, you will need to label them with the appropriate time, culture, dilution factor, and your name. For this time point, you will plate 10 to the 4th, 10 to the 5th, and 10 to the 6th. Make sure that you label along the edge of the plate and on the side containing the media. Do not label on the lid because it is possible to mix up the lids of two plates. Now that you have labeled all of your materials, you may begin the experiment. Remove 1,000 microliters from the overnight culture and place it into the stock tube. Have the P200 micropipetter set to 100 microliters and the P20 micropipetter set to 10 microliters. For the dilutions, remember to vortex before taking out an aliquot of liquid. Pipette 100 microliters from the tube labeled stock and dispense it into the tube labeled 10 to the first. Next, remove 10 microliters from the tube labeled 10 to the first and dispense it into the tube labeled 10 to the third. Now, pipette 100 microliters from the tube labeled 10 to the third and dispense it into the tube labeled 10 to the fourth. Next, remove 100 microliters from the tube labeled 10 to the fourth and dispense it into the tube labeled 10 to the fifth. Finally, remove 100 microliters from the tube labeled 10 to the fifth and dispense it into the tube labeled 10 to the sixth. Make sure that you use a new tip for each step of the dilution. Now that you have finished diluting your sample, it is time to plate the last 2 dilutions. Make sure that the dilution factor on the microcentrifuge tube matches the dilution factor on the plate. Using the P200 micropipetter, Aspirate 200 microliters of a culture from the tube labeled 10 to the fourth. Dispense onto the appropriate plate and repeat this step for the tubes labeled 10 to the fifth and 10 to the sixth. Now you need to spread the cells across the plate. Now that the plates are dry, stack them and take them to the front of the classroom.